Welcome back, fellow scientists. You guys don't get that yet because you haven't done the flipped classroom, but that's how I start all of my flipped classroom videos, which are going to be next unit, regardless of quarantine or COVID-19 or whatever. We would be watching videos for next unit anyways. But that's how I always start all of my flipped classroom videos because, as we talked about in chapter 1, all of us are scientists. All of us are observing our world and asking questions and making hypotheses, even if we don't use those words all the time. We're all scientists. So welcome, fellow scientists. We are going to finish that sheet that I handed out on Monday of the process of meiosis and making gametes. And what I'm going to do, I'm going to use my smartboard presentation that I saved from last year, just because it takes so long to write stuff out and it's messier if I do it on the tablet rather than if I do it on the board. And this way we can go pretty fast. So I'm going to go fast, and you guys can feel free to pause it and write this down. So last year I made the distinction. I'm not sure if I did the vocab the same this year as last year, but we already know diploid, we already know haploid, right? And then we said the meiosis is a process of making gametes, true. But then I made a point last year to contrast that with mitosis, which is cell division, right? And so we can think of meiosis as cell division with genetic reduction, because remember, most of our cells are diploid, right? We have two sets of chromosomes, one from mom, one from dad. To make gametes, we want those gametes to be haploid so that when the two gametes combine, then we get a diploid cell, rather than if both gametes were diploid, then we would get a quadruploid cell, and that just wouldn't survive. That wouldn't be good at all. So we already did this process of meiosis, and we used the same colors. I used the same colors last year as I used this year. So mom, red chromosomes, big A, big B, dad, blue chromosomes, little a, little b. But then last year I made a distinction. Here we go. We'll use purple. Or I made a point rather to show the genotype of this cell. So the genotype would be big A, right? Big A, little a, big B, little b, right? And we would have 4 chromosomes. So our diploid number, the shorthand for diploid is 2n. So our diploid number is 4 chromosomes. All right, sweet. So go ahead, make sure that that's back there. And then let's fast forward all the way. I believe we were at anaphase. There it is, anaphase 1. And so we did our coloring and we labeled the phase. We didn't label what happens yet, I don't think. So just a quick little memory aid. If you remember all the phases of mitosis, right? For meiosis 1, all you do is replace chromosomes with tetrads, and then you got it, right? So in metaphase 1, right, in mitosis, chromosomes line up in the middle. Metaphase, just regular metaphase of mitosis, chromosomes line up in the middle. Metaphase 1 of meiosis 1, tetrads line up in the middle, right? Same thing, anaphase of meiosis, right? The chromosomes split, the two sister chromatids split apart, right? But then in anaphase 1 of meiosis 1, then we just replace chromosome with tetrads, and so tetrad splits, so write that down. And then, of course, notice that the cell starts to pinch in. All right, so the coloring scheme is the same throughout. So you guys can probably just go ahead. They're very clearly labeled, hopefully for all of them. So you guys can just go ahead and fill that out if you want. Telophase 1 and cytokinesis, I kind of combined this because I wanted all the phases to fit on both sides of one page. So same thing as in mitosis, right? Nuclear envelope forms, spindle bundle goes away, cell membrane pinches in totally, right? And then the chromosomes start to unravel, they start to go back. And then that is the end of meiosis 1. So there's nothing really special between telophase 1 and regular telophase in mitosis. All right, so cytokinesis then, obviously, then we have a line. Oops, hang on, I'm still in eraser mode. Then we have a little line right here. Cytokinesis, like the cells are totally split in half. All right, so then we're going to go on. So now here is the start, cell number 8, or picture number 8, is the start of meiosis 2. So that's going to be prophase 2. And notice what we have here. So we have 2 separate individual cells. And how many chromosomes do we have in each cell? Right, well, we have 2 chromosomes. They're butterfly-shaped chromosomes, but they're 2 chromosomes. We started out with 4 chromosomes, remember? So we've achieved chromosomal reduction, but notice because they are butterfly-shaped chromosomes and because the most important thing in meiosis happened, crossing over, our genotype is still big A, big B, little b, right? But now each cell is down to just one N haploid, right? So that's our shorthand for haploid is one N. And so we have 2 chromosomes, two chromosomes. All right, so go ahead, color. Prophase 2, exact same things happen. Chromosomes appear, spindle bundle forms, centrioles move, nuclear envelope dissolves, blah, 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 right? And then, of course, we have metaphase 2, and here it's just like mitosis. So now we don't have tetrads because we don't have homologous chromosomes. We've separated all the chromosomes out. So now we just have our chromosomes lining up in the middle, right? In metaphase 2, same, really simple, really self-explanatory, really straightforward. Anaphase 2, then the chromosomes separate, right? And you'll notice we're kind of continuing our color scheme. So here we had a mainly red chromosome, right, with a little blue tip, and here we had a mainly blue chromosome with a little red tip, right? So then here we're keeping that color scheme, but now the sister chromatids are separating. So this right here used to be a butterfly-shaped chromosome, mainly red with a little bit of blue. This used to be a butterfly-shaped chromosome, mainly blue with a little bit of red, but now our sister chromatids are separated, so now we have individual chromosomes, right? So now for a very brief moment in time, because these cells haven't totally separated, now each of these cells is actually diploid. Why can't I write? So now each of these cells is actually diploid, right? So we have 2N for our cells, so 4 chromosomes, right? Because technically these cells haven't quite split yet, so we have 1, 2, 3, 4, and we still have the genotype big A, big B, little b in each of these cells, right? So then, moving on, right? The next phase is telophase and cytokinesis. So now we're going to pretend that cytokinesis has already happened and there's a line in between there. Telophase, same things happens, nuclear envelope reforms and the chromosomes start to unravel and the spindle bundle goes away. And all the good stuff that happens during telophase, all that happens. Now, watch. Once we have our cells separated, now each individual cell, right now we have 4 haploid cells. So we have 4 N cells, and remember, N, right, equals 2 chromosomes. Haploid equals 2 chromosomes. Right? And now notice, this is so cool. I love this because this explains why if we're looking at 2 traits, remember we said A was something like, oh, I don't know, eye color, and B was something like freckles, right? So we're looking at 2 traits. If we're looking at 2 traits, this is why we have to do a 4x4 Punnett square, a 16-box Punnett square. Because if we start with big A, big B, little b, if we start with this genotype right here, we're looking at one trait, eye color, and then the second trait, freckles. If we're looking at 2 traits, and if we start with that genotype, then telophase cytokinesis, process of meiosis, we get one cell whose genotype is a, little b. We get another cell whose genotype is a, big B. We get another cell whose genotype is big A, little b. And we get another cell whose genotype is big A, big B, which, if you're paying attention, if you remember, right, that's exactly what we would do if we were doing a genetic problem and we had to FOIL this genotype, right? We would do the first letters, so big A, big B, and then FO, right? So do the inside letters, so little a, big B. FOI, we do the outside letters, so big A, little b. And then the last letters, we do little a, little b. So we'd have our 4 different possibilities. This is why, ladies and gentlemen, this is so cool. This is why we FOIL, because the process of meiosis, and most importantly, because of crossing over, increases genetic variability, and so we have our 4 different possibilities right here. So let's take a minute, let's just kind of revel in that and absorb that there's actually a reason why we have to do a 4x4 if we're looking at 2 traits rather than a 2x2 if we're looking at 1 trait. Okay, enough reveling. Let's go on. So, not to muddy the waters at all, but I just also want to point out that meiosis happens differently in males versus females. So, I'm using the human example here. So, the human number of chromosomes in most of our cells, 99.99999% percent of our 37.2, I think it's 37.2 trillion cells are diploid, so they have 46 chromosomes, and most of the time the chromosomes are propeller shaped, right? Only when something is going to go through meiosis or mitosis do the propellers turn into butterflies, right? So we start off, males start off with this. We go through enter meiosis one, right? And so then males make at the end of meiosis one two cells with 23 chromosomes, but they're not propeller-shaped chromosomes, they're butterfly-shaped chromosomes, right? So then we have to do another round of division. So we go through meiosis 2, and then that's finally where we get our haploid gametes, right? Now, if the diploid number, if 2n is 46, right, then the haploid number, single n, is 23, and it's 23 propeller-shaped chromosomes, so that if this gamete right here participates in a fertilization event, then this gamete will contribute 23 chromosomes to the next generation or to the next offspring, right? So that's how it happens in males. This process starts at puberty and it continues on through the entire life. Now, females, it's a little bit different, right? We still start out with 46 chromosomes, right? And then we go through meiosis 1, 23 butterfly-shaped chromosomes, same. But then meiosis 2, right? Something totally different happens is that 3 of these future possible gametes they kind of shrink and they kind of become nothing and they kind of get reabsorbed into the ovary because that's where this process happens. And they become what we call polar nuclei. And there's only one gamete that's actually made for every round of meiosis. Males make 4 gametes for every round of meiosis. Females only make 1 gamete for every round of meiosis. And notice we're still haploid and we still have our propeller-shaped chromosomes, whether it's male or whether it's female. And then females are different in another way as well, is that all of their future gametes, all of their future eggs, go from diploid. They go through meiosis one to haploid before they're born. So a girl, female child, right, is born like just born like 1 minute old. She has all of her future gametes paused at the end of meiosis one, right? So then when females hit puberty, then one future gamete that's paused at the end of meiosis one, per month then is chosen to mature and go through meiosis two and then be released for a possible participation in a fertilization event. And then this whole process stops when the female goes through menopause. So that's one way that, or a couple ways that males and females are different with the process of meiosis. And then at the very end, or at the very beginning rather, I promised you a memory aid for the difference between mitosis and meiosis. And I just remembered that my little picture is down here. I can't see it on my screen, but I guess I should just kind of remember that. So I apologize if I've been referencing things that my face has been covering. I'm still getting used to this. It's still kind of new. So mitosis and meiosis, two very similar words, two very similar processes. People often get them confused. So here's my memory aid that I like. All right, mitosis, it has a T, and so I use that T, and I remember that we start with one cell, obviously, but then it makes 2 cells. All right, and it makes 2 cells with the total number of chromosomes. So if we have a cell up here that's going through, if we have a cell up here that's going through mitosis, right, we're going to make 2 identical daughter cells, 2 identical diploid cells with 46 chromosomes each. If we're talking about the human example, right? So mitosis T makes 2 cells with a total number of chromosomes. Meiosis, it doesn't have a T, and so it doesn't make 2 cells. If you're a male, you make 4 cells. If you're a female, you make 1 cell. Right? So none of those are 2. None of those start with T. So that's great. And it does not have the total number of chromosomes that we started with. Remember, we started with 46, right? Or in the coloring example, we started with 4 chromosomes, right? And then we end in actual humans with 23 chromosomes, or with the coloring example, we end with 2 chromosomes. So it doesn't end with the total number of chromosomes because there's no T. So mitosis, T, 2 total. Meiosis, there's no T, right? So it's not 2, it's either 4 or 1. And there's no T, it's not total, it's half the number of chromosomes. So hopefully that has been helpful. Please email me with any questions. I apologize again for going so quickly, but it's on YouTube. You can pause it, you can replay it to your heart's content. So this is technically supposed to be during a block day. So this is technically supposed to be 80 minutes. So I'm going to give you whatever you have left of your 80 minutes to go ahead and to keep working on the 11.4 SSO, right? Turn that in as soon as possible. Keep working on the Beyond Mendel worksheet that's on OneNote and that's just due on OneNote. And then also, let's see, I also assigned on LMS RenWeb yesterday, or actually today, because I'm recording this on Wednesday, but when you watch it, it'll be a couple days ago. But I assigned on Wednesday some more genetic practice because we just need to get really used to knowing which Punnett square to use, whether it's the 2 by 2, the 4 square, or whether it's the 4 by 4, the 16 square. So go ahead and use the rest of your time to work on that. As always, email me with questions. Hopefully this has been helpful and informative. I am Mr. Leo, and I will see you on the flip side.